I've considered myself a feeder and a bleeder for the last 25 years because I feed people stuff and I bleed them to see what happened to their cholesterol or insulin or whatever. But I am now a feeder and a bleeder and a pooper. Oh my God. We get shit from everyone, and we put it in the freezer and we analyze it. And this is a hot topic in nutrition. So I'm going to talk about that for 40 minutes, and then when I stop, it's up for grabs. You can ask me anything after that. Okay? And I'm an industry shill. I took some money from Beyond Meat, and I'm on the advisory board for a group that I will talk about briefly, but I don't get any money from them. So here's the outline that I have for you for today. I'm going to talk about prebiotics and probiotics, and you've probably seen this. It's in tons of magazine covers, the microbiome. I guess that's why you showed up. And it's in a whole lot of books as well. One at the top left, I'm totally going to pitch. My collaborators, Justin and Erica Sonnenberg, have written a fabulous book called The Good Gut. They ferment everything at home. They make sauerkraut, kimchi, kombucha, sourdough bread, and they have some recipes in this as well. So I don't get any royalties from it. The end vision here, we're not there yet. The end vision is we could diagnose your microbiome, see what kind of species that you're missing that would be helpful, and maybe we can introduce them, or maybe we can give you the foods that would support them, and that this would help not only digestive health, but all kinds of other things. That is our long-term goal, and we aren't even close to there yet. But I do want to say that that is the goal, and it's really fun working with so many fabulous people on campus, so I'm going to highlight a couple of them. Okay, microbiome. So, if you don't know this, the NIH just committed $170 million to 5 years of studies across 5 different centers. 10,000 people are going to poop for all of us multiple times, and then a subset of them, 2,000 are going to poop in a different way, and 250 are going to give extra poop. And they're going to be characterizing all this around the theme of precision nutrition or precision nutrition health. So they have all these centers all over the country that are doing this. Stanford isn't one of them. Actually, we have no nutrition program on campus. My position's with the School of Medicine, the Department of Medicine, which has 14 divisions, and I'm in the Stanford Prevention Research Center, which is kind of like a little mini school of public health. But I teach in human biology, which isn't nutrition, but I actually get a lot of students interested in sports nutrition, child nutrition, nutrition policy, and so that's usually a good home for me. And once in a while, I come to the business school, because there's lots of entrepreneurs that are coming up with new food products that they'd like to try out. If you dig deep into this, or not even that deep, into this big NIH-funded thing, there is a huge microbiome metagenomic center that probably most people don't know about yet, and I think it's going to become more and more common to hear this. I think you'll go to the doc, and you might end up just standardly giving a poop sample along with peeing in a cup and something else. So we'll see how that goes. I'm curious, did anybody spend $90 and already have their poop analyzed? Anybody want to be honest? Yes, okay. No wonder you look so good. That's fantastic. So I'm sure they told you, oh, you have this lactobacillus, and you have this firmicute or Bacteroidetes, and you should eat blackberries, not raspberries, and you should eat walnuts, not pecan. Did they? Okay. At this point, some of it is a little silly. Oh, you're a forager. Oh, you're a harvester. Oh, you're something else. What should I eat? Well, you should eat vegetables and whole grains and nuts and seeds and beans. It's like, that's what they told me anyway, even though I wasn't. All right, anyway, you can spend a lot of money on this. There's a whole bunch of companies coming out doing this. You can send in your poop and pay them. They're actually using your data to come up with bigger data sets to help be predictive of what's going on. And there's quite a few of these. Aravel and Habit are already gone, so some of them don't last. Day 2, which is put together by Eran Segal, brilliant guy at the Weizmann Institute in Israel, is doing some great stuff with this. I hang out with some folks in the UK who sort of have an international collaboration called Zoe. Actually, The last time I gave this talk, somebody was doing the Zoe app. So I'll just ask this time, is anybody in today's talk doing the Zoe app? There we go, right there. Okay. Well, we might ask you some questions later, Kathleen. Okay, and then these guys got in a lot of trouble, and so it's not all going very well. But there's some fraud, there's some good folks out there. We'll see what's going to happen. So my background is it's exciting, it's moving, there's companies, there's innovation and disruption. That's why we're at the business school. But at the basis of all this is really two categories of things, because I'm a nutrition professor. My PhD is in nutrition science, and it's prebiotics and probiotics. So maybe because you came here, you already know this kind of stuff, but I'm going to assume not everybody knows this. Prebiotics is basically fiber. So the definition of fiber is that it's carbohydrate-like molecules in long strings that we don't have the human's enzymes to digest. And so since we can't make it small enough to digest, it goes to our colon. And then the bugs there eat it. And so this is feeding our friends with fiber, so they love this. And the other thing is probiotics, and that's eating live bacteria. So technically, probiotics usually means pills. So technically, fermented foods aren't probiotics, but they contain live bacteria. That's the point. You're eating it. Although not everybody does this. You can take cabbage and then just put vinegar on it, and it might seem like it's fermented, but it's not. I know that Costco now has some kombucha. I might be making this up, so don't quote me. That somebody said they sterilize it and they kill all the bacteria off, and then they add bacteria to it, which seems odd, but maybe from a scaling up perspective, they can't make it the regular way. But I have it at home. I make kombucha all the time. I'm on like my 120th batch right now. It's super simple. It's fermenting right next to my desk once a week. In fact, every Saturday I'll go and I'll make 6 bottles of kombucha, and I'll take my SCOBY, my symbiotic culture of bacteria and yeast, and I'll brew up some tea, and I'll stick some sugar in there, and I'll let it ferment for a week, and then I'll bottle it, and I'll put something in it. I'll put cinnamon and nutmeg. Oh, I should put some pumpkin spice because it's Halloween. I've got some mango extract and some other pomegranate extract, and then I let it ferment a week in the bottle, and I have carbonated kombucha at home. It's pretty fun. Okay, so I make my own probiotics. The Sonnenbergs make way more than me. They're amazing at this. And so from my nutrition perspective, should you eat more fiber? Yes, always. It's like from day one. This is nothing new. Should you eat fermented food? Wait till I show you what we did in our Fi-Fo study. Fi for fermented, Fi for fiber, and Fo for food. Fi-Fi-Fo? You like that? Okay. So I actually stole a bunch of these slides from the Sonnenberg, so I need to give a lot of credit to them. This is Justin's version of your intestinal tract. This is a very dumbed down idea of your intestinal tract here. So up top in the beginning, it's much smaller, and at the end, it's this big colon. And the idea here is that as humans, we have about 20 different enzymes to digest carbohydrates. If you go to the colon, collectively, the bacteria living there have 1,000 different enzymes to digest and break down the fiber-like molecules that show up there. And so the idea being that if we eat the standard American diet, SAD stands for SAD, if you have the standard American diet, which is very high in sugar and refined grain, we are all capable of digesting that. And look at those poor bacteria in the colon just languishing lethargically. They're like, please send me some food. I'm starving down here, because we've broken it all down and absorbed it. But the option is to eat vegetables and legumes and whole intact grains and things like that. And they come with fibers that we can't digest, and so those go down. And look how happy those bacteria are in the colon up at the top there. And while they're digesting that, they are making small molecules. There's a fabulous colleague on campus named Michael Fischbach who got a $7 million grant to study the small molecules that they generate. And I've seen his graphic of 100 different small molecules that get generated by the microbiome that we absorb into our circulatory system. And quite a few of them, we believe, improve immune function and lower inflammation. That's sort of the promise out there. And one of the main types is called short-chain fatty acids. So you might see that a couple times today. It's SCFA, short-chain fatty acids. So altogether, we have two very different metabolic scenarios here. And the Sonnenbergs have sort of jokingly suggested that not all the fibers will feed the bacteria, and it's not all just soluble or insoluble. It's mostly soluble, but it's a bit of a mix. And so what they call the right ones, and we don't actually know exactly what all the right ones are, but they are the microbiota accessible carbohydrates, which they coin as a Big Mac diet. Right? Is that bad? That is so bad. Okay, so now you've seen your intestinal tract. So you don't have a screen. I love this auditorium, but I hate that there's like no screen. So sorry, I'm trying to see that one. I'll look over my shoulder. So, Justin won a photographic award for this electron microscopy picture. So this is a cross-section of a mouse colon stained in various ways. And so if you look in the upper left, those are your friends, the bacteria. So they've stained all the different bacteria in different colors. Isn't that cool? And on the bottom right, they've stained the nuclei of the intestinal cells that line your colon. And so that's the blue is lighting up the intestinal cells. The green that looks bright, dynamic green there, neon green, is the mucus lining that separates the middle of your gut from exposure to the intestinal cells. And this is a very important mucus lining. It protects us from different things. And we want a nice, thick mucus lining. Interestingly, the main composition of the mucus lining is carbohydrate-like substances. In the absence of eating enough fiber, the bacteria will eat your mucus lining. And so this is what they've done in their mouse studies, looking at all the trillions of bacteria that are in there and the way biology works. They fed a bunch of mice, upper left, a whole food diet, let's say, and the poor mouse on the bottom is getting Coca-Cola and jelly beans. If they then track what happens in their colon, the one on the left has a very thick mucus lining. The one on the right has very much of a deteriorated mucus lining, the idea being that if the bacteria keep eating that and thinning it, it will lead to leaky gut syndrome, inflammation, and a lot of problems related to heart disease, diabetes, and cancer. And so this is one of the explanations for the importance of the microbiome and of feeding the microbiome with fiber. So that's the background to this. And then he does some other amazingly fascinating things. So let's go from ecological to some mouse models. So from ecology, he actually has a whole bunch of Hadza poop in his freezer. He went to a traditional hunter-gatherer tribe, and he collected all this poop, and they've been looking at it, and it's amazingly diverse, and it doesn't really look like our Western diet microbiome. They eat a lot of fibers, so they're out hunting, and oftentimes they won't catch anything hunting, and so one of their staples is just tubers. They eat a whole bunch of tubers. And so that's, I think, baobab or something like this on the right. Very, very fibrous. And so we've got a timeline way up top there, and we've got a ton of hunter-gatherer, a little bit of agrarian, and now we've got industrial French fries, which doesn't have nearly the fiber of those folks and what they're eating. And so this is a graph I can't describe because I'm not a microbiologist, but the idea is if you go to all the hunter-gatherer tribes around the world, there's not that many, but they're in different locations, their microbiome is quite similar to one another in different places on the planet. So this graphic is trying to show how they line up from different places in the world, and then if you go to Western countries on this side, they line up also, but in a different way. And so if you look here are 3 different species that they're quite likely to find in a hunter-gatherer tribe, but really exclusively not in Western diets. And there's one in Western diet that's not in the hunter-gatherer tribe. So this is kind of bizarre that there's such commonality around the world for a hunter-gatherer lifestyle and a Western, but they're so different in some specific ways. And the way they liken it to make it more simplistic is this lush rainforest on the left and clear-cut forest on the right is what our intestinal tract looks like. Pretty sad. And so again, timeline here. going from eating hundreds of grams of fiber a day to 35, which is the national guidelines pretty much, to less than half of the national guidelines, which is about what we eat. And my concern has been lately in the nutrition world, this weird backlash against, oh, low fat, oh, it was wrong, low carb, low carb, low carb. And so I got a lot of people doing the low carb thing and eating more meat and eating less fiber. And so I think that's even more of a problem than we've already got. The national recommendations are about 30 to 40 grams of fiber a day. If you look at the National Health and Nutrition Examination Survey, Americans eat about half that. And when they complain, oh, doc, I can't eat that much fiber, it's too much. Oh, GI distress. We kind of want to say, go look at the Hadza. They eat like 100 grams a day. So you can, physiologically you can. You just probably have to assimilate and adapt, and don't do it all at once. Take some time. So here's something you can't do with humans. So I'm a human interventionist. I don't deal with mice. Justin deals with mice. This is super clever. So he took 4 generations of mice, and he tried a fiber experiment with them, where one group across the 4 generations kept getting a high fiber, mixed fiber diet. And the other one would have fiber removed from the diet, and then after a while, added back, as if to say, okay, I came with this fabulous microbiome, I went on the standard American diet, and I lost my microbial diversity. Can I add fiber later and get it back? Would that help if I go back? So here's my 4 generations of mice. So here is pictorially the depth of the color. Over here is how abundant it is, but these are all the different species that they're finding in the mice, if that makes sense. And then they take this first generation, so the next 2 bars are a low fiber diet where you see many fewer species. And then if you can see, I don't really have a good pointer. Maybe I can point real quick, because this is probably too small. See how it says low there and high there. So keep that in mind. So at the end, they give them more fiber. And can you see a few more species come back at that bottom one? OK, now let's go to the next generation. Ooh, even less diversity, but the fiber brings it back. Oh, even less diversity, but the fiber brings it back, but not very much. And then here's the 4th generation. And that's compared to the mice who got fiber the whole time and never lost any diversity. So you can't do this in humans, right? If I was trying to publish this, I'd have to have my great-great-great-granddaughter publish this finding for me. But he does this in mice. And the concern is that, are we depleting the microbiome irreversibly? So a lot of the grants we've been writing lately are sort of titled, here's our grant to recover or restore microbial diversity. Like, we had it, and we're losing it. And so there's some urgency here. We don't want to lose it forever. OK, so hence comes, we actually had a lot of fun. We'd been on the same campus and heard of each other for a while. And the first time we met was at a conference, which actually happens more often than you might think. Like, we're on campus and we don't see each other, but we're both speaking at a conference back to back. And it was like star-crossed lovers or 2 pieces of the amulet that matched. And I said, poop is icky. And he said, humans are a pain in the ass. And I said, we can do this. I will get the humans. I'll get them to poop, and you analyze it. And we can take all this mouse work that you've been doing, which seems to implicate diet, and let's do it in humans. So we got this published in Cell just a little while ago, and I had some great graduate students working on this. We had Dahlia Perlman, who's the diet whisperer, who can get anybody to eat anything. She's so kind and empathetic. And the idea was we would give a bunch of people fermented food and a bunch of people high fiber food, and we would kind of compare. And there's an interesting side note to this, which is all of Justin's work at that time had pointed toward fiber. He did make a lot of fermented food, but he didn't have much faith in it. And he has said that I somehow had to cajole him into including fermented as one of the groups. And you're going to see it proved to be very powerful. So here is the diet whisperer, Dahlia, trying to get people to eat more fiber or more fermented food. It wasn't a huge study. 18 people in a group. For 4 weeks, they ramped up to as high as they could go. And we didn't actually have a set number. We just said as much as humanly possible, just so we can get some movement in your microbiome. And then keep that for 6 weeks. And then we'll go back a few weeks later, and we'll see kind of what you've maintained, if that makes sense. OK, so here is how much fiber they're eating on the left. They started at about 20 grams a day. That's what Americans eat. And check it out. They got higher. And look over here for fermented food. How many of you guys think you eat a serving a day of fermented food? I'd be curious, because our group ate less than a serving a day on average. Anybody eat 2 servings a day? OK, so I'll tell you what we asked them to eat was 6 servings a day. Does that sound ambitious? Keep in mind that a half a cup of sauerkraut or a half a cup of kimchi is a serving. And one of those bottles of kombucha that you buy in a store right now, that's 2 servings. So really, all you'd have to do to get 6 servings a day is a cup of sauerkraut for lunch, a cup of kimchi for dinner, and drink a bottle of kombucha during the day. And it's only about all the different mixtures that we put together, it's about 300 calories. So 6 servings a day is not like, oh, God, that's all they ate was fermented food all day. That was like 15% of their diet. Okay, so it's not as bad as it sounds. Okay, so very cool. Here's what they're eating at baseline. And they ramp up. And then they maintain, bless their hearts. Oh, love this Dahlia Perlman. She got them to keep this. The really interesting thing was at the end, we said the study's over. You can do whatever you want. And I'm most impressed with the fermented folks who were not eating anything in the beginning. On their own, they were eating 3 servings a day when the study ended. They enjoyed it. Oh, my God. Okay? Unapologetically delicious fermented food that we gave them. Okay, and the fiber folks, they're eating more than they did at baseline, too. Something seems to be working. They keep doing it themselves. Okay, so here's the fermented food group. And they are increasing their microbial diversity. That's great. We don't know exactly which ones are good and which ones are bad. So increasing your diversity per se doesn't have to mean it's good, but probably, especially if you're eating well. These are all the different types of things they ate. And statistically, if we teased it apart, it looked like the yogurt and the kombucha were making the biggest contribution. But maybe that's just because what people had the easiest time consuming. We didn't really find that many different fermented foods out there. There's not a huge amount of choice. We did not try to introduce natto. Does anybody here eat natto? OK, oh my God, I cannot. It is so slimy. OK, it is a fermented soybean thing. Let's just not go there for now. So I can eat those, though. And then we found a number of species that seem to be moving in the gut. And this is really exciting. We kind of thought, oh, well, that makes sense. They're eating fermented foods, so they're getting the bacteria from those foods. So we actually started going out and buying all the foods that they were getting. And in the Sonnenberg lab, they characterized all the different species of bacteria that we were finding in those fermented foods. And this is a pie chart of how many of the new bacteria in the increased diversity was contributed from the foods they were eating. So that very small sliver was what they found in the foods. And the rest of the pie was not in those foods that they were looking at. And so the idea here is maybe there's some bacterial strains that are in our gut that are just sort of waiting to bloom, but they need the microenvironment to change. And so we were changing the environment. And so this is the hypothesis we have to follow up on later. It wasn't just what they were eating. Something else, by changing that environment, was opening up the possibility of increasing diversity in other ways. This is kind of how mysterious the microbiome is right now. We don't know exactly what we're doing, but this was fascinating. Okay, and then we have all kinds of outcomes. So we're very interested in immune function and inflammation because this is one of the more likely things that's going to be the clinical impact of these. And so one of the tests that we can do looks at different inflammatory markers. Now, I'm sure you've all been to the doc and had your blood pressure, your LDL and HDL cholesterol, maybe your glucose. How many of you have had your inflammation checked out? Not yet, really? So maybe CRP. There's some docs who ask for CRP, but there isn't. How about your IL-6? How about your tumor necrosis factor? There's so many of them. It's actually a really challenging field because there's so many different inflammatory markers, and we haven't figured out which ones are the right ones. But in the fermented food group, of 90 different inflammatory markers we looked at, and we could have looked at more, of the 90, 20 of them went down during the course of this study. And so this is a little subtle in this graph. So fermented food is in purple, and fiber is in green, and only one changed significantly in fiber. And it was interleukin-8, and it's the last one in my list here. And it's actually anti-inflammatory, and it went up, so that was good. But only one thing got better in fiber, and 20 things got better in fermented food. And Justin called me up and he said, did you mix up the coding? You handed me all the data. I humored you and included a fermented food group because I knew the fiber would do everything we needed. But there's one group that's not really responding here, and the other group is responding in an enormous way. Are you sure you did? Nope. It was the fermented food group that was doing so well. We actually, if you go back about a year, the New York Times Tuesday Science section ran two things. One was our study, and then they got so many requests, the week later, they ran a whole thing on where do you get fermented food, because so many people were curious how we got these results. The fiber group was actually a little different in that, and this is not my forte, but if you do some machine learning type thing, you can see that there were clusters within the 18 people that were on high fiber. And this is pushing the limits of statistics a bit here, and so don't take this too seriously. If we sort of broke them up in thirds, there was one group whose inflammation got better, and there was another group whose inflammation got worse on this study. Whoop, I went too far. And when they dug a little deeper, and this is the hypothesis we're following up on, it was the ones that had the least biodiversity at the beginning of the study that had an adverse reaction to all the fiber. And Justin and Erica's interpretation of this is like a fire hose of fiber that you're not ready for. You don't actually have the bacteria ready to process all this fiber that's coming your way. So in our new studies, we're actually feeding them fermented food first and then fiber to see if we could increase the diversity, and then maybe they can handle more fiber. So at the end of the day, we got this published in Cell, which is a science journal of great reputation. And we said, we got some interesting changes in the fiber. It seemed like more of a personalized response. The response was kind of based on what your baseline diversity was, whereas opposed to, for the fermented, it kind of worked in everybody. Everybody had this very consistent increase in diversity and decrease in inflammation, whereas the fiber was more individualized, which kind of makes for a nice story. There's a very generalizable result on the one hand, and maybe there is something to personalized nutrition. Maybe there is this angle that we should follow up on, that not everybody's the same. Okay, so that's sort of our take-home message from this Phi-Pho study. Have to give a huge shout out to studying immune function and inflammation. So Stanford has a world-famous human immune monitoring center run by Mark Davis and Holden Maker. So anytime we come up with anything that, like if we have $1,000, we could spend it $10,000, we could spend it $1 million. Like if we have more money, they can look more into immune function. So that's a good and a bad thing. The bad thing is, oh my God, we can never run out of extra things to look at for immune function. And we don't have this simple number to just say, oh, you're a 46 and you're a 23. You need to get your number up so it's closer to, yeah, we don't have that yet. But I'm sure everyone's heard of inflammation as sort of this underlying cause of chronic degenerative disease, non-communicable disease. It's there. I just got to figure out how to work it. And there's no one better to work with than these folks. So we feel like we have this trifecta going. We've got a bunch of little studies going where our group intervenes on the diet, the Sonnenberg lab characterizes the microbiome, and the Human Immune Monitoring Center is looking what happens to inflammation. So we're really hoping that Stanford is uniquely poised to take sort of these three fields in an interdisciplinary way and move the science forward. Okay, so on another little note, here's an example. How many of you have had any of these plant-based alternative meats? There's actually a microbiome story here. Okay, so this is the study with appetizing plant food, a meat-eating alternative trial. Not bad, huh? Okay. And so we were looking at something called trimethylamine oxide, which is something that when you're eating meat, it has the precursors choline and carnitine in it. And as that goes to the intestinal tract, the microbiome chews up those two precursors and generates something called TMA, trimethylamine. And if that goes to the liver and gets oxidized, you get trimethylamine oxide. This is sort of an emerging cardiovascular risk factor. It was discovered by Stan Hazen, a doc at the Cleveland Clinic. It's not standard now. It might be one of those things. He would like it to be. I'm very involved with the American Heart Association, and every year when I go, there's a whole session on TMAO and what people found. So you may find this soon, but it's probably related to clotting and inflammation, and lower TMAO is better. Okay. So in our trial, we said, okay, let's have people eat 2 servings a day of fake meat and 2 servings a day of real meat. And we'll have them cross over and do the other one. And we'll use Beyond Meat. This is where I'm the industry shill, so feel free to call me out. I didn't get any money to do this on my own, so they gave me some money. And what should we compare it to? Should I have got McDonald's meat? Should I have got Safeway meat? Well, I'm a huge proponent of not setting up a straw man, so we went to San Francisco, and we got a company called Good Eggs to give us grass-fed, pasture-raised, organic, regenerative, grown meat. So we did the best meat that we could versus Beyond Meat, and we lowered trimethylamine oxide. And we also, in a small way, lowered weight, and we lowered LDL cholesterol. So basically, 3 things got better when they did the alternative meat relative to the regular meat, and nothing got worse. So it was a win for the Beyond Meat. But what I want to focus on is that TMAO right now, and here's that thing I was talking about where TMAO, the precursors, go to the gut transforms it to TMAA, goes to the liver, TMAO contributes to heart disease. That's the whole hypothesis. So in our study, we had 2 orders. So let's start on this side, because half of them did the plant first and the animal second, and half of them did the animal first and the plant second. And so this is a graph of TMAO levels, and you can see, at least on the second half here, when they did the animal first, not everybody, but a couple people, their TMAO shot up, right? Notice that a couple people, it didn't move at all. So it doesn't happen with everyone. Okay, then in a crossover study, the advantage is everybody is their own control, and so that helps to do a study with fewer people. But what you expect is you'll get the same thing regardless of which order it is. And if it doesn't, you are tongue-tied and you have to explain what happened. And so here's the folks who got plant first. You can see the black line is the average, so it goes down a little on average. And then we give them animal, and it oh crap, it didn't shoot up. Now I have to explain this when I try to get it published, and it's not as easy as it was supposed to be. And I did it with fewer people than I maybe needed. But we went to the literature, and we found that somebody had tried to get vegans to eat meat to see if they would make TMAO, and they didn't. And so the idea was, on a vegan diet, they had transformed their microbiome to something that didn't metabolize the choline and the carnitine in the same way. And so right now, we have swap meat poop in the freezer, and it's being thawed, so we can see if in this first phase, they changed their microbiome on that diet in such a way that they didn't make the TMAO later. So these are kind of the fun ways that we get to work with the Sonnenberg lab on how that's happening. So we're going to take advantage of Justin and Erica and their great book, The Good Gut. Again, I don't get any royalties, but it's a great book. Now I'm going to do a shameless plug, because here's another thing that we're doing. How many of you, let's see, probably none of you are pregnant today, but some of you know pregnant people. So here's another acronym from our group, the Maternal and Offspring Microbiome Study we're doing in 132 pregnant women, moms. And the idea is, up front in their second and third trimester, some of them eat more fiber, some of them eat more fermented food, some of them do both. We were trying to learn from that other study. Do both, and some of them do none, so it's the control group. And then what we're going to do is, after they do that for their second and third trimester, then we check out the microbiome of the infant, and we track them both for 18 months. Because one of the issues that's a hot topic now is the mom giving her microbiome to the child. And I don't know how many of you have heard of this, but there's women having C-sections that are freaked out that their kid isn't getting their microbiome. So they're swathing some rags in vaginal juices And then they lay it out over the infant born by a C-section, hoping that they'll, I don't even know if it works, but they're doing that because it seems like, oh my God, I had a C-section and I missed out on this thing. So we're going to have women who have gestational diabetes, C-sections who get antibiotics. We're going to have women who breastfeed or not, wean their children on different food or not. And so the whole hypothesis here is it's actually more discovery and exploratory. How does the kid optimize the microbiome? Are there some dietary factors or other things? So this is another just cool thing. We won't have any results for all. We actually have 65 women already. And so the moms have to give us blood and poop and vaginal swabs and breast milk. And pretty much from the infants, we just need diapers. That works out pretty good if you can tell. OK, and so for this, now we need a clinician, so now we're taking advantage of OBGYN. Dr. Scarlett Karakash is fabulous at this. There is a center on our campus called MCHRI, the Maternal Child Health Research Institute. Fabulous new partners, again, multidisciplinary kind of Stanford research, just perfect. And so now it's the trifecta with a new population to study. Now we're taking all the same things we've been doing before, but we're specifically studying pregnant women. So click on the QR code, or if you know someone who's pregnant, just this is easy to remember, nutrition@stanford.edu. We were originally recruiting women in the Bay Area, but we've expanded it right now to all of California, and it could go beyond that. So during the pandemic, we learned more how to do studies remotely. So we have 68 of our 132 women, and we need the other ones. So hint, hint, if you know. And it's a recruiting plug for our mom study. And then a group that I work with that's also trying to collect data to do this is Zoe. And I saw someone sat down who's doing the Zoe app. This is fabulous. So I get to work with Jose Ordovas at Tufts and a whole bunch of people at King's College London on this Zoe app where they started out over here with 1,000 identical twins. And then they said, oh, let's recruit some people in the US for practice remotely. And they came and sought me out and Jose and some folks at Harvard, and they recruited 1,000 people in the US. And now they have this app, and they're giving advice in the app, and they're trying to get 10,000 people on the app to sign up and give them poop and blood, and just learn from it. So it's a bit of a scam here. I'll be quite honest. It's a bit of a scam. You're paying to get the app, you're contributing your data, and you need that data to figure out what some of this stuff is. So the NIH is $170 million. That's 5 years of 10,000 people, and all that poop they're trying to collect so they can look at it. In parallel, Zoe's trying to do the same thing, but they're actually giving you advice as you go. As they learn, they're developing the app with your data, and they're trying to share that. A lot of it has to do with wearing glucose monitors. So you can see these continuous glucose monitors when you're eating something and your glucose spikes or not. So that's sort of their claim to fame in Zoe. Anyway, these are, I'm supposed to inspire you in these classes without quizzes. So Stanford is doing some super cool stuff, I think, right? So the Zoe group published a huge paper, 2 papers in Nature Medicine just last year. And one was, how much of it is the microbiome versus the genome versus sleep versus exercise versus timing of meal? And then the second paper was a huge deep dive into the microbiome. Which bacteria seem to be the healthiest? Which bacteria seem to be the least healthy? Which ones change? Which ones are associated with foods? I don't possibly have time to share all that with you, but it's very exciting. We have a lot of data, and I don't have a clear message for you today, so I want you to be skeptical of it. I do. I want you to be skeptical, but there's a lot of potential. So conclusion is, please, at least for now, feed your friends. Eat fiber. You can't go wrong. We've been telling you that for years. Here's another reason to eat fiber, right? And then the whole prebiotic, probiotic thing, they have pills for it. Don't have pills. You should eat food. Eat fiber and fermented food. Oh my God, have Korean kimchi. Have Middle Eastern vegetable salad. Have Mediterranean roasted veggies. Make your own kombucha at home. Have a hummus sauerkraut salad. Oh, have this yogurt parfait. Get out there and eat some fiber and fermented food that's unapologetically delicious. enjoy it, and take home these messages. The Western microbiome is not doing so well. Dietary fiber should help feed that. Fermented foods look like they'll help increase diversity, but most of the claims out there right now of companies that are selling this are ahead of the science. They're saying we can do this, and we can't yet. So lots of promise. I think this is where we're headed. I think we're going to be able to diagnose some things, figure out how to support your immune function through the microbiome. But we're not there yet. But I'm glad you came to class today so that you could hear this. And I'd like to give a shout out to all the Sonnenberg Lab folks and all the folks in my lab.